Hello, everyone. Today we're going to focus on some intro to cellular division notes. As always, pause the video as you need. If you need to go forward or back, whatever you need to do, if it's in red, make sure you copy it into your Google Doc. So, the cell theory, you may remember from the very beginning of the course, cell theory is comprised of three things. That all cells come from pre existing cells, meaning cells don't just pop into existence, they came from somewhere. Cells are the basic unit of life, and all living things are made up of at least one cell. So bacteria is alive, it's made of one cell. You are alive, you're made of 3 trillion, give or take a few billion, cells. So these are three tenets or three parts of the cell theory. Keeping the cells identical. So the instructions for making the cell parts are encoded in the DNA. So each new cell must get a complete set of DNA. If you get a scratch and you're trying to regrow those skin cells, the other skin cells around it can divide and they will provide each new skin cell with the same amount of DNA. So what more that looks like is DNA replication. We practiced DNA replication, and then a step further was transcription of translation. DNA replication is solely copying or replicating the DNA before a cell divides. This isn't going into transcription and translation where we read what protein we're making, because the goal of this is not to make a protein. The goal of DNA replication is to make more DNA. So the goal is to copy or replicate each part of the DNA, so then each new cell will have an identical copy of DNA. You can see here in this picture, the DNA strand is unzipping. The base pairs are lining up. A goes to T, because this is DNA, and C goes to G, and a whole new side is forming. So we have a parent cell. Parent just means the original, and then we call the new ones the daughter cells. Why are they daughter cells and not son cells? No idea. There's really not a reason that it's parent cell and daughter cell. That's just kind of what we call it. It's the conventions. Our chromosomes. So our chromosomes are these little X-shaped things. You, as a human being, have 46 chromosomes. You could have one more or one less. But on average, a human being has 46 chromosomes. Some things like a butterfly can have more. Butterflies have more chromosomes than you do, but a fly has less. A fly only has 8. Bacteria, going on that, have 1 chromosome. If you remember when we talked about bacteria, bacteria do not have a nucleus. So they don't protect their little chromosome. It's just out in the center, floating around the cytoplasm. And that is why bacteria mutate frequently, because they do not protect their DNA and it can get messed up very, very easily. So the DNA inside of a prokaryote, like bacteria, is just one big circular chromosome. Now, eukaryotic chromosomes, like your chromosomes, your dog's chromosomes, a plant's chromosomes, they store genetic information in your chromosomes. And like I said, humans have 46 chromosomes or 23 identical pairs. So you have 2 of each. So you have a chromosome 1 and 2 of those. Chromosome 2, 2 of those. And we'll do more about that later. But you have 46 total. Now, each chromosome is composed of a whole bunch of DNA molecules. Chromosomes can't be seen when cells aren't dividing. And when you just have all this like DNA scattered everywhere, we call it chromatin. Chromatin is just uncoiled DNA material. Chromosomes are the little X shapes they're made into when we're trying to replicate our DNA and replicate our cells. Just like if you were to go on vacation, you would pack, but normally your clothes are scattered everywhere in closets and drawers. But when you go on a vacation, you would pack all of your items into a suitcase. That way it's easier to move them. Same thing with cells. So when we compact the DNA into our chromosomes, we use these proteins called histones, and that helps wind the DNA up so that it stays relatively straight and we don't have a situation like when you put your headphones in your pocket and all the cords get all jumbled up. Yeah, we try to wrap them around things to prevent that. So chromosomes in a dividing cell. Duplicated chromosomes can be called chromatids, and are held together by this word called the centromere. So the centromere is the center, and each half of the X shape is called a chromatid. Sometimes they're called sister chromatids. Again, parent cell, daughter cell, sister chromatids. It's just, it's not that they actually have like a sign gendered with them. It's just what we call them as a convention. So I wouldn't think much about it. A karyotype would be a picture of someone's DNA. You can actually go and get your DNA spread out into a karyotype, and then someone very carefully lines up the pairs. So you see chromosome 1, both chromatids together. Chromosome 2, 3, 4, 5. And then if we get very down to the bottom, we have, let me move B, we have an X and a Y. So looking at this person would be assigned male at birth because they have an X chromosome and a Y chromosome. If you are female at birth, you have an X and an X. There are, of course, crazy conditions of being XXX, XYY, XXY. Chromosomes are weird. We'll talk more about those. So the first 22 pairs, all the way up here, are called autosomes because they're just chromosomes that make you you. It deals with your skin, your hair, your eyes, your height, your heart. But the last one is that last 23rd pair are the sex chromosomes. So like I said, in general, XX female, XY male in general. There's a lot of conditions and exceptions to that rule. So the looking at the chromosomes, this is kind of the fun part. The X chromosome is not the same as a Y chromosome. The X chromosome codes for color blindness. If you have color blindness, it's on this big X chromosome. The X chromosome has information on it. The Y chromosome honestly just kind of codes for testosterone. So if you have a Y chromosome, it will have your body generate testosterone. That's about all it does. It does have a few things on it, but look at the size difference. There's not much genetic information on there. All right, let's talk about cell reproduction. So how do we make new cells? There's different types of cell reproduction. Asexual, A kind of means not. Asexual reproduction is where one cell divides to make two new daughter cells. So asexual does not involve any sort of mixing of DNA. It's basically to make clones. Mitosis and binary fission are examples of asexual reproduction. Mitosis is one of your cells, like a skin cell, might just, divide itself and make new cells. You don't need your skin cells to mix genetic information because they're just making skin cells. Sexual reproduction involves two cells, like an egg and a sperm, joining to make a new cell, which we call a zygote, that is not identical to the original cells. Sexual reproduction involves mixing of genes. Meiosis is the example there, and we will spend a lot of time talking about the differences between mitosis and meiosis. Now, in prokaryotes, so in bacteria, right? Binary fission, I mentioned binary fission on this slide, is an example of asexual reproduction. Oh, there we go. Binary fission would just be one cell, it grows larger and then just pinches itself in half, breaks in half. Now you have two cells. And that is binary fission. Would be really crazy if humans divided that way. We don't, which is great. Now, the cell cycle is the life cycle of a cell. Every living thing has a life cycle. You have a life cycle, your dog has a life cycle, and your cells do too. For the most part, cells are not immortal. Most of their time is going to be spent in this gray part interphase, which is G1, S, and G2. I'll give you more on that. And then some of their time is spent dividing. But your heart cell has to have time to be a heart cell. It can't constantly be dividing or it wouldn't have time to do its job. So the first phase is G1. G stands for growth. So G1 is the first growth stage after cell division. The cell's maturing, it's making extra cytoplasm, it's making organelles. So G stands for growth, and there's two of them. So this is G1. All that time we spent talking about DNA and DNA replication, DNA is replicated in the S phase. S stands for synthesis. So the synthesis stage is where DNA is copied or replicated. Replicated is the fancy word for copied. So you have your original DNA, your replicated DNA. They're going to be identical. G2 is your second growth phase. So this occurs after you've copied your DNA, all your cell structures are made, and you're also making more organelles, more mitochondria because they're the powerhouse of the cell, and some more proteins are being made. At each of these phases, they're double checking to make sure they did it correctly. So the G1, did it grow correctly? Okay, yes, keep going. S, did we make enough DNA? Yep, okay. Then G2, did we grow? Do we have all the organelles we need before we finally get into this last phase, the mitotic phase, where we're going to do mitosis? So most of the cell's cycle is spent just being a cell, an interphase. But there is a wedge of it that is talking about division, right? So we'll talk more about mitosis in the next set of notes. But the whole point of this was just to kind of see that the cells aren't constantly dividing. If they are, that's something that does lead to cancer. But the cells normally just spend their time being a cell, doing their job until they hit that phase for mitosis. And that is something we'll talk about another day. So feel free to go back, get anything you missed, and I hope you have a wonderful day.